Okay, recording has started. Can everyone see my screen?
Yes.
Yeah. Okay, so a very quick recap. What we have been doing for the past two classes. So we started talking about the biophysics of action potentials. We talked about the structure of a neuron and how a neuron passes information to another neuron. Um, so we talked about the passive properties, the fact that the ionic concentration across the membrane is different, and there are active mechanisms, pumps that maintain this concentration gradient. Because of this, uh, the ion is at equilibrium only when the potential is at some particular value, which is called the equilibrium potential. This is one when there is only one ion in the picture. When there are multiple ion channels that are open, then the cell is at resting state, in which none of the ions are actually at equilibrium, but the potential of the cell doesn't change because the total sum of currents is zero. Okay, so this is what we got to get to the resting membrane potential. And then we talked about the fact that there's a capacitor as well. So therefore, in a passive condition, the cell just behaves like a simple RC circuit with which causes it to charge and discharge in an exponential fashion if you inject some current in it. This was the first class where we talked about passive properties. Then in the previous class, we talked about the fact that in actual neurons, this is not what really happens. The neuron is very non-linear, so that once the potential reaches some threshold value, it leads to the generation of what is called an action potential. Okay, and this comes about only because the ion channels are their conductance is not fixed, but it depends on the voltage across the membrane. So some very clever experiments were done by Hodgkin and Huxley to find out what these conductances are, how they change, and they did it on this squid axon by putting two electrodes and then measuring the potential and actually holding it to a particular value. The current that would be needed to do that was provided by this second electrode called the current passing electrode. And then you could measure the current that are flowing through the membrane when you hold the potential at a fixed value. Okay, this is the voltage clamp technique. And Hodgkin and Huxley found out that when you do that, the current changes in a particular way, and you can break it down to the constituent sodium and potassium current. And this shows that the sodium conductance and sodium ion channels in general open up very quickly and then close, giving this sort of a conductance profile, while potassium ion channels slowly open and stay open. Okay, and if you put this together, this is the part that I skipped, where it shows the same thing at the level of a particular ion channel. But anyway, so the macroscopic and microscopic stories are similar, so there's not much there. This is the structure of a sodium ion channel. It has two types of gates: the activation gate that opens quickly when you depolarize the cell, and an inactivation gate which slowly comes and closes the ion channel, and then the sodium channel is inactivated, and it will only get activated when it comes back, when the potential comes back to the membrane potential, resting membrane potential or below. Okay, and if you put all of this together, you can actually explain the generation of action potential, which is what we did in the last class. Okay, And then we talked about the fact that because the sodium channels are already inactivated, if you pass another pulse of current, it won't lead to another action potential for a while. And this is the time that it takes for the sodium ion channels to get back from the inactive state to the resting state, where this ball has again come out and it's the other gate that is closing it. Okay, so this is called the refractory period. Okay, so this is where we stopped in the last class. If there are any questions on this, I'll take them. Otherwise, I'll move forward. Okay.
Sir,
So
actually,
let's move.
I'm able to see the shared screen. Is the screen already shared?
Yeah.
Okay,
Are
okay.
others able to see the shared screen?
Yes, sir.
Yes, sir.
Okay. So yeah, others are able to see it. So I don't know. Maybe
Okay,
you
sir,
can log
I'll
out and
fill it
log
up.
in. Yeah. Okay. So I'll move forward then. So well, there isn't much here really. So this is just how an action potential passes from one side of an axon to another. So if you have got an axon and you're recording the action potential at different locations along this, you will find that the action potential magnitude is the same everywhere. Okay, it's just that the time at which it's generated is delayed. So it's close to 1.5 milliseconds here, and it's close to 5 milliseconds there. So, but there's no reduction in the amplitude of the action potential. Therefore, the action potential can actually pass for arbitrarily long distances. Okay, even if the axon is like a meter long, there won't be any attenuation in the voltage. It's just going to get generated at a later part after some time. Okay, and the reason why this happens is very straightforward. So suppose you are stimulating at one part. So you stimulate at one part of the axon. So the sodium ion channels open up because of this depolarization. Sodium current comes in, membrane gets depolarized, and this process leads to an action potential. So there's a lot of positive charge momentarily inside. Some of that is going to move along the length of the axon, thereby depolarizing the nearby portion. Okay, and now when the nearby portion is also depolarized enough, there's going to be a new action potential generated at this point because the sodium ion channels here are going to open, leading to an influx of sodium. So the potential is now positive here. Second, I'll change to laser pointer. Yeah. And now this will again lead to current that flows along both directions, this direction as well as that direction. But notice that even though the potential here is more now, the sodium channels are still inactivated. So the current on this side won't lead to another action potential, but on this side, it will lead to an action potential. Therefore, the direction is actually unidirectional. If you stimulate, it's going to lead to an action potential on this side. Of course, if there's axon on that side as well, then there will be another action potential. But usually you have the cell body right here. So when when an action potential is generated at one point, it moves in one direction. It just doesn't keep going crisscross because the ion channels are inactivated, and it takes a while for them to get back to the resting state. So that's it. That's how an action potential moves. So nature has played more tricks. There's what is called myelination. So you cover part of the axon with what is called myelin. It's like like a duct tape, which which prevents current to move out. So the current actually just keeps moving inside the cell. And then there are these little gaps. They're called nodes of Ranvier. And in these gaps are packed with sodium channels. So once more current comes in, it leads to an action potential here. So it action potential actually jumps from one node of runway to another. Okay, it's called a saltatory motion. Saltare means jump in Latin. So once you myelinate the axon as compared to an unmyelinated axon, the action potential can travel very fast. So it just jumps while the unmyelinated one just moves continuously. So it's much slower. So this is how action potentials move from one part of an axon to another. And eventually, when they reach the end of the nerve terminal, the action potential leads to the release of neurotransmitters. This is the part that I don't cover in this class because it's another involved set of procedures. So people are waiting in the lobby. So I can't select this when I'm in this mode. Trying to reach to this meeting in general, but I can't select it anymore now that I have come to this laser pointer mode. Arrow lets me do it. Something new that I learned. Okay, so somebody was in the lobby but has managed to come in. Please come on time. Yeah, please come on time, everyone. Otherwise, sort of disrupts the flow a little bit. As I said, I'll try to finish right on, start right on time at 9 o'clock, finish by 9:55. So please come on time. Okay, so sorry, I sort of lost the thread there a little bit. The myelinated axon jumps from one node of runway to another. Unmyelinated axon is slow. And in fact, you can show that if you look at the conduction velocity as a function of fiber diameter, it increases as a square root of the diameter in case of unmyelinated fibers, but it increases linearly for myelinated fibers. So this is basically what nature did in order to speed up the action potential. It basically started myelinating the axons. So you actually don't need very thick fibers, nerves, in order to move the action potential quickly. Okay, so this is the end of the biophysics part. So we are finishing this. As I mentioned, the part where it reaches the end of the nerve terminal and releases neurotransmitters, I am not covering that. You have to just believe me that once it reaches the end of the nerve terminal, there are these little pouches called synaptic vesicles that have the neurotransmitter in them. They fuse to the membrane and release the neurotransmitter. These neurotransmitters then bind to the receptors that are on the postsynaptic cells and open those ion channels. And then again, the whole cycle starts. Again, when you open the ion channels, the sodium comes in and it leads to depolarization of the next cell. And if it's strong enough, it leads to an action potential there. Okay, and then it moves on from one neuron to another. And that's how information is carried from one part of to another. Okay, so that's that. And now we are going to move to the next part of the class. So any questions on the biophysics, or can I move on?
So,
yeah.
In the previous slide 42, could you explain why these two graphs again? I'm slightly confused.
So actually, I didn't talk about how conduction velocity is determined in case of first passive case as well as active case for unmyelinated and myelinated. So it's another so the passive case is what we do in great detail in the other class, but it takes about 1.5 classes to do it. It turns out the neuron, the axon in particular, in the passive case, behaves just like a cable, a cable for which the cable theory can be used. It's the same theory that electrical engineers use for cable line transmission. This is I'm talking about electrical transmission from a power grid. It's the same math, with the exception that biological tissue doesn't have inductors while your cable lines do. So you can work out that math, and people have done that. It's very, very cool. In previous years, we used to actually do a little bit of that in this course as well. But you can actually prove that if in a passive case, the conduction velocity depends on the square root of the fiber diameter. Okay, so I'm not doing it, so that's why it's a bit confusing here. When you come to active mechanisms, it's a little bit trickier, but people have done that. This is in fact work of Hodgkin himself, but the proof is harder there. So I don't do it in this class or the other class, but this is something you'll have to just again, either read up on your own or just take on faith, because the math here is more involved. But people have worked out that the conduction velocity, as you increase the fiber diameter, increases, but then the increase is following different formulas. So in one case, it's a square root for unmyelinated fiber as well as a passive fiber. For an active fiber, it becomes linear. Okay, so you'll have to just, as I said, take it on faith, or I can refer to you the references that do that, and you have to read that up, or take the other course where we do that in a lot more detail. Here, I don't want to spend too much time on the biophysics because it's a signal processing oriented course. So you're just getting a brief sort of overview of certain things. Okay.
Yes. Okay. Yes, sir.
All right. So now let's get to.
So just a clarification, while like finding the fiber diameter for a myelinated neuron, you include the myelinated portion or like?
Yeah, that is why it doesn't start from 0. So that's why it's, that's the myelin length. Yes.
Okay, yeah. Thank you.
Okay. So let's start with the second part now, which is our understanding of random variables and correlations. So, okay, so let's get to that, and then I will tell you sort of the way this, we introduce certain topics. So now we are in the techniques part of the module. We've finished the biophysics part. So in the next 3 odd classes, this is what we'll be doing. And these are the reference in case you are really interested, but the level at we are going to deal with these topics, even the lecture material here is sufficient. So here's how we will do things. Okay, so for each module, I'm going to give you some data and some MATLAB codes. This is a very MATLAB heavy course that way. I'm going to show you some data, and then I'm going to ask some questions. Okay, so that's where the application comes in. These are questions that are deeply buried in the neuroscience literature. These are very sort of classic fundamental questions that neuroscientists ask. And then we are going to just develop the math that is needed to answer those questions. So we'll be developing stuff selectively, because a comprehensive development is just not going to be possible. We'll be just stuck with spikes for the entire course if we do that. So at times you'll be feeling that there's some parts that are sort of not as comprehensive, and that's there for a reason, because learning to do learning this way also has its advantages, because you can always see the application and the motivation why certain things are being done. So actually the best way to do this course is to also have some formal background in the math that we are going to develop, but it's not necessary. So if you are also just starting out, it's okay. We will develop whatever is needed to answer the questions. Okay, so now let's get started. I'm going to introduce you first to the data. So this data set that I'm going to give you is collected from a monkey's brain by inserting a microelectrode array, which looks like this. So this is a surgery in which this chip is implanted in the brain of a monkey. It's like putting a pacemaker, so it's once the surgery is over, there's only a little connector that sticks out of the monkey's head. The monkey is otherwise perfectly fine. In a week or so, they recover and then they do the task that they are trained to do. And you can record neural activity from about 100 odd neurons from the monkey's brain. Okay, and then in our case, the electrode is implanted in the primary visual cortex, area V1, which is at the back of the head. And the monkey does a task in which a couple of stimuli are shown on the screen. So the monkey is positioned in front of a screen, and the monkey looks at the dot at the screen, and you present two stimuli, one of which falls on the receptive field of these neurons. What is a receptive field? A receptive field is nothing but the visual space that elicits a response from these neurons. If I had positioned my electrode at a different part of the brain, the receptive field would have moved. In fact, different brain areas have receptive fields of different sizes. But in the primary visual cortex, the receptive fields are small, which means that each neuron only sees a small part of the visual world. And it's quite amazing that these millions of neurons that are in the visual cortex sort of stitch together this information to create the world that we see. Okay, so each neuron is actually seeing a very small part of it. So a typical experiment is like this. When you present something on the screen, like a bar or a dot of light, that excites certain neurons in the retina. From there, it goes to a structure called a lateral geniculate nucleus, or LGN. And from there, it goes to the primary visual cortex, which is where our electrodes are. Okay, so these are the electrodes that I talked about. And interestingly, turns out these neurons in the primary visual cortex are actually sensitive to very simple features about the stimulus. For example, its orientation. If I have a light, a bar which is horizontal, for example, so this this axis is time here. You are presenting the stimulus for a short duration, about a second or so. If you present a horizontal bar, the neuron doesn't care. But if you present a vertical bar, the neuron fires a bunch of action potentials. Here, the action potentials, because they are very fast events, are represented as vertical ticks. Okay, so the neuron would fire, let's say, 6 action potentials when you present a vertical bar, but not a horizontal bar. Now, if you record from its neighboring neuron, you will find that that neuron likes 45 degrees. Another neuron might like 90 degrees. So the different neurons that we record from prefer different orientations. Okay, this is in primary visual cortex. Likewise, if you present a spot of light at the center of the receptive field, you see an on response, which means that the neuron fires. Action potentials when you present the stimulus. At the edge of it, once you move a little bit away, you see a different type of response whereby the neuron responds once the stimulus stops. There's this YouTube video here that very nicely shows this. I used to show it in class in previous years. This year I'm going to skip because it's kind of hard to see it, do it online. But I really encourage you to see this. It will give you a feel of how these recordings are. If you actually present uniform light throughout the receptive field, you barely get any response. So these neurons are very selective to spots of lights and bars. Okay. And as we go from one visual area to another, the responses become more and more sophisticated. So in your area V2, which is after V1, neurons care about contours, for example. In area V4, they would depend on curvatures, whether it's sharp or oriented in a particular way. If you go to area like IT, neurons respond to faces, or very complicated, like dogs but not cats, on Bill Clinton but not Hillary Clinton, and so on. So neurons become more and more selective and more sophisticated as we go up the visual hierarchy, and all of these areas get activated within like 200 milliseconds, so that if I show you an image of Amitabh Bachchan, within 150 milliseconds, you can actually recognize the face, even though the receptors that are carrying this information only see little spots of Amitabh Bachchan, little bits of Amitabh Bachchan at the periphery, and it all gets stitched together. It's actually incredible how sophisticated our visual systems are. Okay, yeah, so this is what I was telling you about the orientations. If you look at a piece of visual cortex here and look at what orientation they prefer, they are color coded this way. You see that there's a very beautiful mosaic sort of a structure, where now if you zoom into a little bit, these are called pinwheels. You've got some a center here that is not really orientation selective, but around it are these these columnar structures which prefer different orientations, and they are aligned in a very sort of nice pinwheel structure. This is the structure of the visual cortex, orientation selectivity in visual cortex. There's also selectivity for spatial frequency, selectivity for which eye is information is coming on. It's called ocular dominance, and a bunch of other things. Okay, well, back to the data now. So the data is available. So it's there on our course web page.
Sir, in the
Yeah.
grouping of in that pinwheel structure, in the grouping of neurons for specific orientations or any other features, I think the grouping will vary for different types of features. Like it's not like all
Yes.
the
Yes.
yeah.
Yes. So
Okay.
you've got what these are columns, and then you have got a hyper column uh, where you have all the different features orientations together. And as you are saying that the grouping for ocular dominance actually looks different. It's bigger than the, so its orientation is more finely uh, uh, grained than ocular dominance. Spatial frequency is also different. There are different types of receptors. Parvocellular and magnocellular, they have different spatial frequency preferences, and then there is some separation at the level of LGN, but then it goes to, so it's very complicated. Okay, I can't really get to it. It's a it's a research career in itself. People have spent decades figuring out the orientation, different preferences, and how they are aligned and how it's all put together. So yes, to give a short answer, what you are saying is true, but it's quite complex and quite involved, so we won't get into too much detail here. Okay.
Yeah. Yes, sir. Thank you.
Okay, so if you go to this course web page, NS212, you will find this resources here. It's a zipped file, and it's called Module 1 Resources. If you download this, you will get to the data and the codes. So all you have to do is then start MATLAB and go to that folder, and then just use the commands that are written here. If you just run this command, it will generate these plots. Okay, it's a quick way to get started on MATLAB. MATLAB is interestingly not a program that many of us formally learn. It's a program that you learn just by playing with it. Okay, it's a very simple and very intuitive program. The only thing is that you have to really remember that is that when you are not sure, just type help and the command name, and it will tell you how it's how it's doing things. So help plot will tell you what plot does, help xlim will tell you what xlim does, and so on. Okay, so if you haven't seen MATLAB, please don't be sort of afraid of it. Especially if you haven't learned it formally. Most of the people that I know of, including myself, never learned MATLAB formally. It's not usually something that you learn formally. You just play with it and eventually it will start making sense. It's very intuitive that way. So that's what we are going to do. I'm going to just write these MATLAB codes. Please do this, okay? So don't postpone this. It's actually a lot of fun to have the data and play with it as well. So please spend some time doing it, and you'll see it will be a very fun and rewarding experience. So you download the data, you load the data file. So these are called MAT files, MATLAB data files. Load command loads this MATLAB data file, and it generates this data structure called spike data, which essentially has times at which spikes were generated for 76 repetitions of the stimulus. Okay, so what did I do? I showed this monkey a stimulus 76 times. Not one time, two times, but 76 times, one after another. I showed the monkey this stimulus. Okay, so in a typical task, the monkey stares at the screen for 2-3 seconds. You show, 4-5 stimuli during that time with some interstimulus interval. And then if the monkey is able to maintain fixation or, in an attention task, detect some change in the stimulus, the monkey gets some juice reward. You see that juice pipe here, the monkey gets juice, the monkey gets happy, and the monkey continues to do the task. And then you record the neural data while the stimuli are being presented. So the data set that you guys have this stimulus that's presented 76 times. So this y-axis is trial number, and the stimulus is presented for 400 milliseconds. And the times are with respect to that time. So time 0 is when a stimulus is presented till 400 milliseconds. And then for 600 milliseconds, there's no stimulus. Okay, so minus 600 is when the previous stimulus was switched off, and then for 600 milliseconds, there's nothing. I'm only showing you between minus 0.4 to 0.8 by using this command, which limits the x-axis to minus 0.4 to 0.8. And then between 0 to 0.4 is where the stimulus is presented. So the neuron is responding by firing more action potentials. See that? There's more action potentials. But it's note a couple of things. One is that it's not that the action potentials are generated at exactly the same time. As you move from one trial to another, for the same stimulus presented at the same time, 0, the response is slightly different. And the second thing to note that even though the stimulus is the same between 0 and 0.4 seconds, there's some variation. Initially, the onset is, causes a large change in firing rate. So the neuron responds and then it sort of settles down. Okay, so the firing here is not random, but stochastic. We'll make this term clearer. Okay, so there's another command called PSTH plot. PSTH is peristimulus time histogram. It just counts how many spikes are there in some small bin, and then let's say 10 milliseconds, and then therefore plots the firing rate. In general, how many spikes are there on average? So it's just like the average of all these spikes over some small pins. So this is a very typical response of a neuron. The neuron, even when there is no stimulus, does fire some spikes, but it's low. So we'll go, generate a few spikes. When you present a stimulus, there's a huge increase in firing rate, and then it sort of settles down to a lower value, and it will remain like that until the stimulus is removed. Then it again goes back to baseline firing. Okay, there's some delay, as you can see, about 30-40 milliseconds in this case. And this is the time it takes for the information to reach from the retina to the primary visual cortex. As you move to V2 and V4 and IT, the latency will increase a little bit. And that's just because it takes longer and longer to reach that brain area. Okay, so this case, you have the data and you can actually see how one neuron fires action potentials when you present the stimulus. And in fact, I've given you data from two neurons. So if you put spike data list 2 here, you would see the firing rate of a second neuron as well. Two simultaneously recorded neurons when the data was presented 76 times. Okay, so that's the data. Now here's the question. So this is the format that we are doing. I'm just first showing you some data. Then I'm asking you some questions. Then we'll develop the math to answer the question. So the question is as follows. And this actually is a profound question that people have been struggling for the last 30 odd years and still there is no answer. So the question is as follows. Here, if you look at the firing, you can see that it's not the same, right? From one trial to another, if I look at trial 1, the spike trains look in a particular way, so action potentials are generated at certain times. And if I look at trial 20, it's something else, trial 40 is something else, even though it's the same stimulus being presented 76 times, the responses are slightly different. So the question is, which aspect of this firing is carrying information about the stimulus? And there are two schools of thought. One is that it does not matter exactly what time a spike is generated. What matters is the number of spikes that are generated in a given time interval. Okay, so if I think of information it's coded in, let's say, 50 milliseconds, 0 to 50 milliseconds. So in each trial, I will just count the number of spikes. So let's say 200 milliseconds. So I'll have 1, 2, 3, 4 spikes in the first trial and 8 spikes in the second and 3 and so on. But the point is that the only metric of interest are the number of action potentials that are generated within a time interval. It doesn't matter when those action potentials are generated in that time interval. This is called a rate code. Okay, so the rate coding scheme, precise timing of the spike doesn't matter. Only the metric of interest is the total number of spikes. As opposed to that, there's a temporal coding scheme in which the precise timing of spikes is also important. And there's a reason behind that, because you see neurons, a bunch of neurons from V1 will be connected to one neuron in V2, right? And if V1 neuron fires a lot of action potentials, that may cause the V2 neuron to also fire. So the V1 neuron has carried its information to V2, but also if a bunch of neurons start firing together in synchrony, then their neurotransmitter release is also going to be synchronous, and it will be easier for neuron in V2 to respond to this synchronous firing. So the temporal coding scheme has its advantages. Maybe you can synchronize the firing rate and then convey the information to V2 versus a case in the rate coding scheme where you are just increasing the firing rate. Okay, are the coding schemes okay with everyone? Rate code versus temporal code?
Sir, I just feel these definitions kind of, I don't know, seem to overlap just depending on what you consider as your timing interval, right? So, like if you take a very fine time interval,
Yeah,
that's what
that's a
becomes a
very
temporal
good
code.
point. Yes, if you increase the timing interval, at some point, they become sort of one and the same. So we'll consider a long enough timing interval, let's say 50 milliseconds for now. And as we will see, this answer will also become relevant. Having said that, basically, I am saying that if I consider a particular time interval, um, let's say 200 to 400 milliseconds, right? So here the firing rate is about the same, and it's about 20 spikes. So in a 200 millisecond interval, I'm going to talk about 4 spikes on average, right? 20 spikes per second means 4 spikes in 200 milliseconds. So I can make this question very precise, and for that for this more precise question, there is a very clear difference between a rate and a temporal code. The precise question is that if I look at this 200 millisecond window, is the number of spikes in this window irrespective of where they occur? Is that the thing that carries information, or whether the precise timings of the neuron action potentials within this interval also carrying information? Okay, so okay, so is this clear then? Rate versus temporal code within a sufficiently long time interval?
Sir, I had a question about the 76 trials. The thing is, like there's a learning component. Isn't there a learning component
No,
involved
there's
too, sir?
not a
Like,
learning component, because the monkey has been doing this task for months before we implant the array. So there's no learning there. But
okay.
you can also learning by doing this while the monkey is doing the task. And people do that also. Here, the monkey is already learned. So there's no learning component.
Okay, okay. Okay, thank you.
Okay. So, so are the questions okay? Rate code versus temporal code. So now I ask the question, given this data set, tell me whether this neuron is firing randomly or not within, let's say, 200 to 400 milliseconds. Or in other words, is it following a rate code or not? Okay, if it's a rate code, then that means, it will fire 20 spikes or 25 spikes, but it's going to be random. There's no logic to when the spikes will occur. So how will you answer this question? All the information that you need is already here. This thing carries all the information. There's no other information. So let's have a few guesses here. If I give you this data set, which I have given you, and I ask you a question, is the neuron firing, following a rate code? How are you going to answer the question?
So the underlying distribution, I guess, gives me information. So when I look between
Okay, so what do you mean by distribution?
So can you go to that blue plot again, the one with the average, I think. Yeah. So over here, what I see between the 200 millisecond to 400 millisecond almost seems to have a, what do you say, horizontal line that can be fit through it. Whereas if I look between, like, the just,
Yeah. So sure, we're just considering 200 to 400. So
yeah.
it's a horizontal line. Yes. Is the neuron firing randomly in that period or not?
I guess you'll have to do some statistical kind of test, but I mean, visually, it seems to me that it is being picked up from a uniform random distribution. So,
Okay, so what is how do you say that visually? I don't get it. So even if all the spikes occurred at exactly at the same time, right? I guess you would say you would see some spikes there. Is that what you're referring to?
Yeah, so if you were to look at, I guess, look for any kind of outlier in particular, in the sense if there's any point which has much higher than a few standard deviations, you will not find any such point over here because they all seem to be very close to a certain central line. Whereas if you go close to that response just after zero, over there you can see that the spike is quite noticeably higher compared to the surrounding areas. So you can look at this with standard deviations, is what I'm saying.
Okay. Well, okay. So let's, any other, so you're in the right direction, but the logic is a little bit off. We'll get to that in the few classes. Any other ideas here? So somebody said we can see whether it's flat or not flat and the standard deviation around it. As I said, it's going to only tell you a little bit, but not much. You can have what is called a non-homogeneous Poisson process, which could have any shape and be completely random following a Poisson process. So that actually won't get us too far. The standard deviation will help, as we will see. Any other ideas though, before we actually go to a Poisson process?
For this interval, this is following the rate code because it's as it has been pointed out also that if you average it out only from point 2 to point 4, you'll see that it's around it's nearly a flat curve uh, in the in this interval with the like 20 around 20 firing 20 spikes per seconds.
Yeah, so let me just answer why that is not the case. Okay, uh, so imagine this is the firing rate profile, and you guys are saying, oh, maybe it's following a temporal code at around 100 millisecond because look, it seems to change so much, and then it follows a rate code. Actually, the concept is quite different. So see, you can always pick a small interval, let's say, zero point 0.2 to 0.21, some small interval where the firing probability, and let's say we divide this into um, uh, 10 milliseconds bin, let's say. So what is going to be the firing probability here? It's 20 spikes per second. So in 10 milliseconds, you're getting what, point 0.2 or 0.02? 20 by 100, 10 milliseconds, 0.2, right? So in a 10 millisecond window, you can get a spike with a probability of 0.2. Okay, and I can actually say that I'm going to flip a coin and not flip a coin here. In this case, I will choose a random number between 0 and 1. And if it's less than 0.2, I'll put a spike. Otherwise, I won't. Okay, so the firing probability is going to be 0.2. Everybody gets that, right? So at 0.2, at 0.2 seconds, between 0.2 to 0.1, 0.21, I'm going to flip a coin and, sorry, not flip a coin here. We can do it in the coin flip way also, but let me just do it in this random, uniform way. So I'm going to choose a uniform number between 0 and 1. So it could be anything between 0 and 1 chosen uniformly. And if it's less than 0.2, then I'll put a spike. Otherwise, I won't put a spike. Okay, so the neuron between 0.2 and 0.21 is following a completely random rate-like behavior, right? Because it's coming from a completely sort of, whether you will have a spike at 0.2 seconds or not will vary from trial to trial in a completely random way. right? And I could actually do the same thing at each of these bins. So what do? I divide my 0 to 0.4 second into 10 milliseconds intervals, find out the firing probability, which is going to be like 0.1 here, it's going to be almost 0.16 here. It's going, sorry, so it was 0.01 here, 0.16 here, 0.02 here, and so on. But the point is that at each of these intervals, I am flipping a coin, essentially doing a random experiment there. So the neuron is actually going to be following a rate code everywhere in this case, even though the mean firing rates are varying. Just because the mean varies doesn't tell you anything about the randomness in the distribution. It's a tricky point, and we'll need to develop the math to do this. We need to get to a precise definition of a random variable in order to do this properly. But basically, to give a proper formal answer here, it is completely possible that this neuron is firing in what is called a Poisson-like manner, but it's not a homogeneous Poisson process. It's an inhomogeneous Poisson process. Now, some of you may be hearing this for the first time would be very confused. What is a Poisson process? And that is what we are going to get towards in the next couple of classes. And we'll do this from scratch. So even if you haven't done anything about random variables, don't worry, we will get to this. This is the question that we have, and we are going to answer this properly. But just the shape here, the fact that it's changing in this time and is flat here, doesn't mean that it can't be a Poisson process everywhere. Okay, in fact, it could be, as I said, an inhomogeneous Poisson process everywhere. And we will see that this neuron is actually appears to be Poisson-like, but it's actually not. Okay, so let me then just introduce the, any questions on this? This was actually sort of like a preview of everything that I want to do in the next 2-3 classes. So let me just start off by, yeah.
Just a slight random thought, or like just intuition. So the random occurring of spikes can be related with the refractory period itself.
Yeah, so the refractory period doesn't really, so this neuron is only firing 20 spikes a second.
Okay.
And the refractory period is a few milliseconds. So it doesn't really matter that much.
Okay.
If the neuron is indeed firing 500 spikes or something, then you would start worrying about refractory period.
Mm-hmm.
Okay.
So,
So we need a way to define randomness. Okay, so we need to be more precise about what it means to fire randomly in order to answer this question, whether it's firing following a rate code or not. So I'm going to now define a random variable in order to define randomness. Okay, and that's what we are going to do in the next few classes. So just to start things out before time gets up this class, we have what is called a random experiment. Oh, by the way, how many of, well, I guess we can't do a show of hands here, right? I just wanted to know, many of you may have heard of this term random variable, right? Maybe we'll just start this from the next class. Can you guys tell me what do you mean by a random variable? Anyone who may have heard of this term?
yes, sir. So a random variable is essentially just a variable that takes the values from the sample space of the experiment.
Okay, so it's a variable that takes the value from the sample space of an experiment. Is that it? Anything that does that is a random variable.
Like its domain is the sample space and the range is a Yeah, I mean you define it on a sample space and then it takes a value in the real real number set.
Okay, is that it? Any mapping from the sample space to the real line is a random variable.
Uh, it's it's a function.
It's
The
a
Yeah,
range,
it's a
function.
function.
I
Yeah.
think the
Huh?
range have to be between 0 and 1. So you have a very strict kind of
Uh,
definition.
no,
No.
it's
No,
not.
that's
That's
not.
for the yeah.
Okay, no, the range doesn't have to be 0 and 1. It can take any number from on the real line. But is any mapping from the sample space to the real line a random variable?
I mean, generally, one value in the sample space will correspond to one value in the real line. It's not a does not have to be a bijection as far as I.
So if the mapping from so okay, so you guys are right that a random variable is a mapping from the sample space to the real line. So if a random variable maps all the elements of a sample space to the same number on the real line, then is it a random variable?
I mean,
So
we
could you
could.
repeat that?
Well, the question is if the random variable, which is a function from the sample space to the real line, maps all the elements of the sample space to the same number on the real line, then is it a random variable?
No, they have to be different numbers. For like each outcome in the
Uh,
sample space, it has to be a different number.
like by definition, it is defined as a function. So then we define a probability function
No, no,
corresponding to
let's
it,
not
and
get to
yeah.
probability yet. Do you need to have a probability? Well, let's not get to probability yet. We will get to probability. Right now, I'm only asking you about random variables.
Yes, sir. So we could say that it is a random variable that takes the same value base irrespective of what's in the sample space. It's
Okay.
I mean, we could say
So
that.
good. So let me just say we are out of time. Some of you may be just wondering what is a random variable. So we will take this up. None of the answers that you guys have given is correct completely, although partially the answers are correct. So let's do one thing. Let's all of us read a little bit. I'm talking about Wikipedia here. Just read about random variables on Wikipedia. Okay, or one of the basic chapters of Sheldon Ross is rather involved. You could try Papolis. But as I said, uh, read about what is random variable on Wikipedia. We will start from there. Okay, and we will actually get to a precise definition of a random variable. Then we will find out that when something is truly random, the firing is truly random, then the random variable takes a certain form. And then we are going to check whether our data set, in our data set, um, the firing rate does take that form or not. That's our sequence to answer the question. We'll do all of that in the next class. So please read up and we'll pick up from here. So we are stopping at a very exciting stage, so to speak. So please just read a little bit and we will then, go from there. Okay, on the course web page, I will now today put the PDF so you can also read up and get a preview of what is going to come in the next classes. And I'm also going to update the online links. Second one, I haven't done that. So that one and today. So over time, this is just going to build up. Also, please do read this paper. If you haven't read too many papers, it takes a really long time to read a paper in the beginning when you're first starting out. And we will be discussing this paper. All the stuff in this paper is directly relevant to the stuff that we are learning right now. Okay, so that sort of cohesively puts everything together. Okay, so we're done for today. We will start from here, from the same, yeah.